Good morning, everyone. This is Sir Franz, and we will be discussing at this time the treatment of neurodegenerative disorders, in particular, the pharmacotherapy of these disorders. And for part one, we will be talking about Parkinson's disease. Here are my references, and the main reference would be Goodman and Gilman's Pharmacology. First, what are neurodegenerative disorders? These sets of disorders are characterized by progressive and irreversible loss of neurons from specific parts of the brain. So it is progressive, which means it initially starts at a partial state and continues to progress over a long period of time so that at the terminal stages there is frank neurologic phenomena. But at the initial stages, there might be subtle manifestations. Next, the loss is irreversible. Up to date, there is currently no clinically approved therapy that will be able to reverse the pathology that happens in neurodegenerative disorders. And that stems from the fact that neurons are highly specialized cells, which, as they are more highly specialized, prevents them from regenerating. There are particular regions of the brain affected in neurodegenerative disorders. For example, Parkinson and Huntington disease will affect the basal ganglia. Alzheimer's disease will affect neurons of the hippocampus, which would be the deeper and more medial parts of the temporal lobe, where new connections and synaptic processes are formed, um, which subserve the function of memory, and the cortex, in particular the neocortex. So the loss of neurons in the hippocampus and the neocortex underlie the manifestation of Alzheimer's, which is memory loss and the loss of executive function. In amyotrophic lateral sclerosis, there is unique motor neuron loss in the spinal cord, the bulbar portions of the brainstem, and the cortical motor neurons, which are located adjacent to the lateral fissure. Another feature of neurodegenerative disorders would be proteinopathy. There are particular abnormalities in the production and the elimination of specific proteins in these sets of neurodegenerative disorders. For example, in Huntington, there is the preponderance of the huntingtin protein. In Parkinson's, you have Lewy Body. In Alzheimer's, you have tau proteins. Okay, and so these accumulations of proteins would mean that the cell, the neurons in these particular regions, are less able to degrade these proteins and prevent these proteins from accumulating and interfering with neuronal function. Next, there is selective vulnerability. What does this mean? This means that particular neurons in specific regions of the brain are more sensitive towards the effects of the neurodegenerative processes than other neurons. So what could entail this selective vulnerability? Examples would be differences in the susceptibility to excitotoxic injury. Excitotoxicity is produced by the neurotransmitter glutamate, and the neurons in the brain have different sensitivities to the injury caused by glutamate. For example, neurons in the cortex would be more susceptible than neurons in the nuclei of the midbrain, for example. Next, there is regional variation in the capacity for oxidative metabolism. What does this mean? Neurons in the central nervous system have different capabilities in performing ATP production via the electron transport chain. And so, for example, if a patient is exposed to anoxic conditions where there is very low or absent oxygen in the blood, neurons in the cortex would be more sensitive than neurons, for example, in the spinal cord. Okay? Next, there are differences in the production of free radicals by the cellular byproducts of metabolism. And so there will be neurons which produce more free radicals at a quicker rate than, let's say, other neurons. And therefore, the former sets of neurons will be more readily affected by these neurodegenerative disorders. Next, Huntington's disease is exclusively familial. So that means if you have encountered a patient probably with Huntington's disease, there might be partial expression or full expression of the Huntington's disease in his or her parents or siblings. Contrast that to Parkinson's disease, Alzheimer's disease, and amyotrophic lateral sclerosis, where the patients usually present sporadically. That means they will present, for example, with signs and symptoms of Parkinson's disease, but upon probing the family history, you will not be able to elicit any other family member which has Parkinson's disease. Next, there is the propensity to develop neurodegenerative diseases if a patient has, for example, infection of the central nervous system structures, exposure to toxins such as heavy metals and pesticides, or acquired brain injury in the setting of traumatic brain injury. So that means individuals who regularly, as part of their occupation, get head trauma will develop neurodegenerative disorders much quicker. That is why boxers are notoriously diagnosed uh, in higher prevalence than the general population with, for example, Parkinson's disease. Okay, as is the case of Freddie Roach. Next, approach to therapy. There are many ways to attempt to treat neurodegenerative disorders. The first would be the neurochemical approach. We have to understand that because there is a certain dropout of neuronal populations in neurodegenerative disorders, these neurons will not be able to produce um, their natural neurotransmitters. And so the absence of the neurotransmitters would subserve the presentation of the patient. Okay, so the goal of therapy in the neurochemical approach would be to provide or replete this neurotransmitter system so that even if the neurons that naturally have produced these neurotransmitters are already non-functional or diseased, there is still the presence of these neurotransmitters. So just replace and compensate the damage to the neurotransmitter systems. Next would be neuroprotection. neuroprotection aims to modify the underlying neurodegenerative process. But to date, there is still no clinically effective agent that will reverse the neurodegenerative process. But there are evidences that some medications and some practices slow down the progression of the neurodegenerative process. So an example of the use of neuroprotection would be to inhibit the activity of glutamate, which also decreases excitotoxicity, as is what we see in patients who have Alzheimer's disease who are given drugs to inhibit glutamate, such as memantine. Next would be to help the cell perform ATP generation or cellular metabolism in a more efficient way. For example, the provision of the vitamin CoQ10, which is a mediator of the electron transport chain, is purportedly found to slow down neurodegeneration because there is less um, production of free radicals, because the electron transport chain becomes more efficient in a system that has a lot of CoQ10. Okay. And lastly, immunotherapy. So the use of antibodies which specifically bind to the abnormal proteins that accumulate um, theoretically will prevent the some aspects of the pathophysiology of neurodegenerative disorders. This is seen, for example, in Alzheimer's disease, where there is a new clinically approved antibody that binds to the amyloid proteins in Alzheimer's disease. So let's shift gears and focus our attention to Parkinson's disease. For us to understand what happens in Parkinson's disease, we need to review the physiology of the basal ganglia. First, the basal ganglia is a misnomer because these sets of neurons and cell bodies of these neurons are found in the central nervous system. And so it would be more appropriate to call them basal nuclei, because again, in neuroanatomy, they are found at the base of the cerebrum surrounding the diencephalon and the ventricular system. So they are there, they are part of the central nervous system. Okay, whereas when we use the word ganglion, we are referring to cell bodies that are outside the central nervous system. That is, they are in the peripheral nervous system. But because in the olden times they used the term basal ganglia, it has become convention even in medical literature. The basal ganglia is involved in the scaling of strength of muscular contractions. So you have to understand that the primary pathway which connects the brain to the muscle peripherally would be the cortico spinal tract. So that would involve neurons of the pyramidal system, whose cell bodies are found um, in portions of the cortex just adjacent to the lateral fissure. So if that is the main tract that subserves contraction of skeletal muscles, you also have to understand that initially the signal that goes into the muscle would be very crude, and it would not be fine-tuned, so that without the influence of the cerebellum, the thalamus, and the basal ganglia, the amount of force produced by uh, the by the muscles in response to stimulation of the corticospinal tract would be excessive. Okay. Okay, and so that would not fit the goal of fine movement. So the basal ganglia is there because it helps to fine-tune the amount of force required with the amount of force produced. It collaborates or helps the supplementary motor area in organizing the sequences of cell columns in the motor cortex. So again, the pyramidal tract would be found just adjacent to the uh, lateral fissure. So the areas of the frontal lobe cortex contain neurons that attempt to organize the sequence of activation of cells in the primary motor cortex. So think of it this way, that the neurons that are found in the motor association areas would prepare the actual motor cortex by arranging the sequence of steps and arranging um, the force, the order of muscular contraction so that the particular goal of movement is achieved. So together with that function of the association motor areas, the basal ganglia helps in that organization so that there is coordinated movement and the patient does not experience ataxia. The basal ganglia come into action after the corticospinal tract has already been activated by premotor areas, including the cerebellum. So what does this mean? The cerebellum, together with the supplementary motor area, will actually, in some way, preempt the movement and the discharge of the motor cortex. So they will stimulate the motor cortex to fire, and once the motor cortex has already fired and the signal has traveled the corticospinal tract, as it descends into the spinal cord, those signals continually become modified by the basal ganglia. So by sequence, the sets of neuronal structures would be, for the production of movement, the supplementary motor area or the motor association area, together with the cerebellum, which in a way thinks about the movement, the motor cortex, which actually is connected somatotopically to the muscles of the body, and then as those information descends along the corticospinal tract, the basal ganglia modifies the movement or the firing. Okay. So how does that happen? So what you see here would be the motor loop of the basal ganglia. Number one, corticostriate fibers activate neurons in the striatum. So first, let us orient ourselves with the picture. This would be the cerebrum, and in particular, this would be found in the frontal lobe. Okay. Of course, the surface of the cerebrum would be the cortex, and that is where you will find the cell bodies. So neurons from the cortex would actually descend along tracts and integrate with the basal ganglia. So let us first orient ourselves with the structures of the basal ganglia here. You have here the striatum, which is made up of the caudate and the putamen. Then you have the globus pallidus, which is further subdivided into the globus pallidus externa and the globus pallidus interna. and they are intimately associated with the thalamus. So remember, in your ARAS, ascending reticular activation system, the thalamus would play a central role in activating portions of the cortex. Okay? So keep that in mind, and uh, keep the basic neuroanatomy in mind. Superiorly, you have the cortex, and inferiorly, you have the basal ganglia with portions of the diencephalon, the thalamus. So how does the motor loop look like? The cortex will activate the striatum, and the striatum would integrate with the globus pallidus, and the globus pallidus would integrate with the thalamus, and the thalamus would then, based on the total uh, or the final set of information that it receives, either stimulates the cortex or does not stimulate the cortex. Okay, so that is the motor loop, because you start with the cortex and you end up with the cortex. So how does this motor loop actually look like? Neurons from the cortex will stimulate the striatum, and then the striatum, neurons here, would then stimulate or inhibit the globus pallidus interna, and the neurons in the globus pallidus interna, depending on the pathway, would either stimulate or inhibit neurons of the ventrolateral nucleus of the thalamus. And the neurons from the ventrolateral nucleus of the thalamus will either activate or inhibit the supplementary motor area. Okay? So that would be the motor loop. So again, cortex, striatum, globus pallidus, thalamus, cortex. Cortex, striatum, globus pallidus, thalamus. So that would be your motor loop. Let's then go into the generalizations. The cortex, the neurons that come from the cortex, would release glutamate. And since glutamate is an excitatory neurotransmitter, the next neuron would be activated. Next, the neurons here in the basal ganglia, in particular the striatum and the globus pallidus, are small spined, thin medium neurons, which would produce GABA. And since GABA is an inhibitory neurotransmitter, the neurons that come in contact with the neurons from the striatum and the globus pallidus would be inhibited because they receive GABA as the information. The last generalization would be neurons coming from the thalamus also secrete glutamate and acetylcholine, which are excitatory neurotransmitters. Therefore, neurons after the neuron from the thalamus would be activated. So, let us begin our discussion of the direct and the indirect pathway for the facilitation of movement here in the motor loop. Let's begin with number 1. Corticostriate fibers activate GABAergic neurons in the striatum. So, corticostriate fibers, because this synapse releases glutamate, would mean that the neurons from the striatum, the cell bodies in the striatum, would be activated. But because the cell bodies here, again, because they are from the striatum and the globus pallidus, produce GABA, this will be activated and cause GABA to be released onto the next neuron. So let's proceed to number 2. Activated striatal fibers, this one, would release GABA into the neurons found in the globus pallidus interna. So what does that mean? If this neuron receives GABA, that means this will be inhibited. And therefore, this will not fire GABA. Okay? So if the neuron of the globus pallidus interna does not fire GABA, what then happens? The neuron of the ventrolateral nucleus of the thalamus will be activated. Particularly, it is disinhibited. It is prevented from being inhibited. And so it will fire. As the neuron of the ventrolateral nucleus of the thalamus fires, it will then be able to activate the supplementary motor area and therefore facilitate the planned movement. Okay? So that is the motor loop. Now let's key in the function of the substantia nigra pars compacta. The substantia nigra is a neuroanatomic structure that is found particularly in the midbrain. And it is called substantia nigra because grossly the portion is a little bit dark because it contains a lot of dopamine, which later on becomes melanin after further oxidation. So let's factor that here. This is the neuron of the substantia nigra pars compacta. This will release, as what I have discussed a while ago, dopamine. But in the striatum, there are two types of dopamine receptors, dopamine 1 and dopamine 2. Because dopamine 1 receptor is excitatory, the substantia nigra pars compacta, as it releases dopamine, will cause this pathway to be excited, as if it is excited by the cortex. Okay, so what happens? Dopamine here is released, and the neuron of the striatum becomes activated. And because this releases GABA, the neuron of the globus pallidus interna is inhibited. Okay? Once this is inhibited, the neuron of the thalamus, the VLN nuclei, will become activated and stimulate the cortex. So that is a plus point for movement. Think of it as an on pathway. Okay? So that is the direct pathway of the motor loop. Next, there is also an indirect pathway, which relies on the communication of the striatum and the globus pallidus externa. So what happens in the indirect pathway? The substantia nigra pars compacta releases dopamine into the striatum. But this time, the receptors in the striatum, in the striatal neuron, is the dopamine 2 receptor. The dopamine 2 receptor is inhibitory. Therefore, signal, which is dopamine from the substantia nigra pars compacta, will inhibit the striatal neuron. If this striatal neuron, which releases GABA, is inhibited, this will not fire. And so, the globus pallidus externa is released from being inhibited. And so, it will fire. The globus pallidus externa also produces GABA. And so, the globus pallidus externa neuron releases GABA into the subthalamic nucleus of Maynard. This one, the STN. So, the STN will become inhibited by this neuron, which is now active. Okay? So, the STN is inhibited. Therefore, the STN, the subthalamic nucleus, will not fire acetylcholine. Okay? Without the firing of acetylcholine, this neuron in the globus pallidus interna will not be activated because there is no release of acetylcholine from the subthalamic nucleus. Okay? If the neuron here in the globus pallidus interna is not activated, it will not release GABA into the thalamic neuron. And therefore, the thalamic neuron will still be stimulated. So that is your indirect pathway because it has to pass through first the globus pallidus externa. Okay? So at the end of the day, movement is facilitated. However, we now go into the pathology. What happens if the neurons of the substantia nigra pars compacta die out of degeneration? There will be no dopamine. Therefore, there will be no facilitation of the movement. Okay? So let's talk about Parkinson's. There are, in essence, two general types. You can have idiopathic Parkinson's disease, where it is usually sporadic. But you also have a rarer form of Parkinson's, which is the familial Parkinson's disease. Okay? So, in the sporadic Parkinson's disease, there is usually these features: bradykinesia, slow movement, slowness, and poverty of movement. Again, because the supplementary motor area does not receive facilitatory signals from the thalamus. There is muscular rigidity. There is resting tremor, which uniquely abates or decreases when there is voluntary movement. And so we can call this resting tremor because the tremor would appear whenever the patient does not will to move. Okay? Then there is impairment of postural balance, so that later on in the advanced stages of the disease, the patient will lose balance and will have difficult gait. Okay? And so they would tend to fall because of this loss of postural balance. What is the pathologic hallmark? If we take a section of the midbrain and look at it under the microscope, what will we see? There will be loss of pigmented dopamine neurons of the substantia nigra with consequent appearance of intracellular inclusions or abnormal proteins known as Lewy bodies. Okay, and these are made up of alpha-synuclein. So this is the normal midbrain, where you will have a portion that is dark, and this would be the substantia nigra. Okay, and this is rich in dopaminergic neurons. However, in a midbrain of a patient with Parkinson's disease, the substantia nigra is depigmented. It means it lost its dark coloration because the dopaminergic neurons have died from degeneration. Under the microscope, this is what a Lewy body would look like. Okay, so these are your supporting cells here, and this would be an entire neuron cell body that is a little bit swollen, and you have here the accumulation of the abnormal protein, and this is what you call the Lewy body. Next, there will be neurofibrillary tangle in the nerve cell of substantia nigra as seen in post-encephalitic Parkinsonism, seen in progressive supranuclear palsy, and Parkinsonism dementia complex. So these are your Parkinsonian plus syndromes. Okay, but no need to really learn that one, I just want you to appreciate the tangles here that are found in the synapse. And this will, of course, prevent efficient neurotransmission in the synapse. Kasi technically, naaano sila. They become trapped. Okay? And there is turbulent flow of extracellular fluid in the synapse because of the tangle. So again, what is the pathophysiology? There is loss of dopaminergic neurons of the substantia nigra pars compacta, which removes facilitation of movement by the direct pathway. So there is no facilitation of movement, and that leads to poverty of learned movement. There is an overall inhibitory state produced by the disinhibited activity of the lenticular nuclei. So if you will recap the circuit, you will understand that if you lose the dopamine that goes into the striatum, there is no inhibitory activity to the globus pallidus. And therefore, the globus pallidus will then inhibit the thalamus. And when the thalamus is inhibited, there is no stimulation of the cortex. So this is a great picture by Dr. Netter showing to us the stages of Parkinsonism. Stage 1, you have unilateral involvement and you have the characteristic blank fascie. Okay? The affected arm is in semi-flexed position and this has the tremor. Okay? And patient leans to the unaffected side. In stage 2, the disease already manifests as bilateral involvement. Okay? With early postural change, so they will usually hunch over, and they have a slow, hypokinetic, shuffling gait with decreased excursion of the legs. So that is consistent with disease of the basal ganglia, which also has a somatotopic organization. Okay, so next, in stage 3, there is pronounced gait disturbance and generalized disability. So the patient will lose activities of daily living. So because of the postural instability, the patient will have the tendency to fall. In stage 4, there is significant disability and there is limited ambulation already. And in stage 5, there is complete invalidism. The patient is confined to the bed or the chair, cannot stand or walk even with assistance. So let's now go into the meat of the discussion, which is a little bit short because it's straightforward. Treatment of Parkinson's disease. You have to take note that there are no treatments to date that will slow down the neurodegenerative process of Parkinson's disease. We have a little bit of evidence for rasagiline, which is a monoamine oxidase type B inhibitor. But again, that will have to be proven by randomized clinical trials. When do we start treatment of a patient with Parkinson's disease? It is recommended to start the treatment if there is already effect on the patient's activities of daily living. The drugs that we use to treat Parkinson's disease usually belong to three mechanisms. Number one, they can be dopamine precursors, which would mean these are drugs that when given into the patient would become dopamine inside the body. Next, dopamine agonists. These are drugs that look like dopamine and therefore when given to the patient will be able to bind to the dopamine receptors and produce the effects of dopamine. And again, these two would fit into the neurochemical approach to therapy. where we replace the missing neurotransmitter. And lastly, inhibitors of dopamine degradation. The body has a unique way of degrading dopamine so that it does not accumulate in the synapse. But in a patient with Parkinson's disease, where there is scarcity of dopamine, we can give drugs that will prevent the metabolism and the elimination of dopamine so that dopamine stays in the synapse for a longer period of time. So for early disease, we usually use monotherapy. But in the later stages of the disease, we can combine these agents. So monotherapy with levodopa, which is a dopamine precursor, non-ergot, which means these are newer agents, non-ergot dopamine agonists, and MAO-B inhibitors, monoamine oxidase B inhibitors. So in this image, we are shown how the body acquires the precursors for dopamine and eventually transfers those precursors into the midbrain, where you have here the substantia nigra, where the pertinent metabolism for the production of dopamine occurs. And you have your dopamine, which is continuously fed by the substantia nigra pars compacta, the nigrostriatal pathway, into the striatum, which is composed of the caudate nucleus and the putamen. Okay, so dopa taken up by surviving nigral neurons, converted to dopamine, and released at the synaptic junctions in the corpus striatum. Okay, so from tyrosine, you have a hydroxylation reaction, an addition of an OH to form dopa. And then from dopa, you have a decarboxylation reaction, removal of COO to produce dopamine. And eventually the dopamine that is produced here travels the pathway and is sent into the striatum, which is made up of the caudate and the putamen. Okay, so the neurons will then move forward as depicted in our image a while ago. Okay, so tyrosine from the diet can come here into the blood and reach the midbrain. Okay, but this will also serve as the common pathway, the systemic circulation, by which the medicines that we give actually become dopamine in the brain. So we can give, for example, oral levodopa, and that levodopa goes into the bloodstream. Okay, and some of the levodopa will actually flow through the bloodstream and enter the midbrain, where it will be used to form eventually dopamine that is in the central nervous system. However, in the blood, there is the presence of two enzymes which metabolize dopa. You have dopa decarboxylase, which will convert the levodopa in the blood into actual dopamine into the blood. And because dopamine is already very water soluble, it will not be able to pass through the blood-brain barrier. And so the dopamine that is formed in the blood will not actually reach the brain because it has been activated or converted readily and prematurely in the blood. Okay, so a little bit of dopa that enters the brain will be the only amount of dopa that becomes dopamine. Okay, that dopa that is in the blood will also be metabolized. The dopamine metabolized by monoamine oxidase and catechol O methyltransferase, which will decrease the levels of dopamine and dopa that reaches the midbrain. Okay? And so later on, as we discuss the pharmacotherapy and the pharmacologic classes, we will revisit this idea. First, you have dopamine precursors. Essentially, they are dopa. These drugs are analogs of dopa, dihydroxyphenyl acetate, where the dopa that is in the body naturally is decarboxylated. You remove a COO to form dopamine. And so the goal of treatment with dopamine precursors would be to supply dopa into the patient so that the patient will utilize the dopa and form dopamine. Exogenous levodopa is given and is taken up by the central nervous system. In the midbrain, it will be decarboxylated and it will generate dopamine, which is the neurotransmitter missing in Parkinson. However, the levodopa can be decarboxylated prematurely in the blood, where the dopamine formed can produce unwanted systemic adverse effects, such as instability of the cardiovascular system. So what do scientists propose that doctors do? Carbidopa should be given so that this drug will prevent peripheral metabolism of the levodopa so that there is a higher level of levodopa that enters the central nervous system and there is a lower level of dopamine in the blood. Okay, so let us illustrate that here. The levodopa that is given to the patient penetrates the blood-brain barrier and it becomes dopamine in the midbrain. However, if the levodopa is given alone, this will be converted by the enzyme dopa decarboxylase into dopamine in blood so that the dopamine will produce the adverse effects, which is cardiovascular instability. So what do? We give a drug which is carbidopa, which is structurally similar to levodopa, so that the carbidopa will be taken up by the dopa decarboxylase and inhibits the dopa decarboxylase enzyme, so that the dopa decarboxylase enzyme does not attack the levodopa, which will be spared for uptake into the midbrain and converted into dopamine. Okay, so that is the principle with the use of combination carbidopa and levodopa. Carbidopa technically is a suicide substrate. It will offer itself up to dopa decarboxylase so that dopa decarboxylase does not attack levodopa. Next, we have dopamine agonists. These are drugs that look like dopamine and so are able to bind to dopamine receptors in the midbrain and in the striatum and produce the naturally associated effects of dopamine, which is the facilitation of movement. The older agents are derived from a fungus, which is ergot. Okay, so this is a fungus that grows on wheat. So the fungus, which grows on wheat, produces substances, alkaloids, that look like dopamine. Okay, so that the older agents or older drugs that we use as dopamine agonists come from this fungus. Newer agents, however, are chemically synthesized. They are more potent and produce less adverse effects, which again would be in the cardiovascular system. Ergot alkaloids are notorious for causing hypertensive emergency. Examples of ergot-derived agents would be bromocriptine and pergolide, and non-ergot derivatives would be ropinirole, pramipexole, and rotigotine. Pergolide is an intermediate between the two. Okay, so look at this one, the image on the right. The healthy synapse, which contains dopamine. You have normal signaling. But in Parkinson's disease, there is a relative lack of dopamine, and that leads to impaired signaling, and this will manifest as bradykinesia and the manifestations of Parkinson's disease, the four cardinal manifestations. However, even if you only have a little dopamine in a patient with Parkinson's disease, if you give the dopamine agonist, which would be these orange circles, you will still be able to occupy the receptors and produce some semblance of the normal signaling with dopamine. So you have improved signaling. And lastly, we have inhibitors of dopamine metabolism. They will prevent methylation of dopamine. The enzyme that adds a methyl group, CH3, to dopamine would be catechol O methyltransferase. That methylated dopamine is already inactive. And so, what do? We give drugs that will inhibit catechol O methyltransferase and therefore prevent the methylation of dopamine so that dopamine stays as dopamine and will be able to bind to its receptor. Drugs include tolcapone and entacapone, which can be used as monotherapy in early Parkinson. We have other drugs that prevent oxidative deamination. So dopamine has an amine group, an NH3 group. So if you remove the NH3, that will inactivate dopamine. So that process of removing the NH3 is oxidative deamination. You oxidize the dopamine, and in the process of oxidizing the dopamine, it cleaves the NH3. So that is a way by which dopamine is inactivated. Therefore, we can give drugs that will inhibit the enzyme that removes the NH3 so that dopamine stays as dopamine for a longer period of time. The enzyme that removes the dopamine via oxidative deamination, that removes the amine, would be monoamine oxidase B inhibitors. Examples would be selegiline and rasagiline. There were studies around 2015s which found out that rasagiline actually in a substantial portion of the participants of the study slowed down the progression of Parkinson's disease. But of course, that would have to be tested with a lot more patients. They thus will prolong the action of dopamine in the synapse. And as our summary slide, we have this, which will show the sites of actions of the different drugs. First, let us orient ourselves. This would be the blood side, and this would be the brain side, and this is your blood-brain barrier. The levodopa, which is the drug that we give as a dopamine precursor, would be taken up by the amino acid transporter in the blood-brain barrier and therefore will be available into the brain. The dopa here would undergo decarboxylation to produce dopamine. And dopamine would be the one that binds to the dopamine receptor and produces the effects on the direct and indirect pathway. So what do we do in the presence of neurodegeneration of dopaminergic neurons? We can give drugs that will be able to bind to the dopamine receptors and produce the effects of dopamine. These are your dopamine agonists: bromocriptine, pramipexole, and ropinirole, the newer agents, except bromocriptine. Next, we can prevent the degradation of dopamine via methylation by inhibiting the enzyme catechol O methyltransferase so that dopamine is not methylated and stays as dopamine. Examples of catechol O methyltransferase inhibitors would be tolcapone. Next, dopamine will also be degraded into dihydroxyphenylacetic acid or DOPAC via oxidative deamination. So we can prevent the degradation of dopamine into DOPAC by inhibiting monoamine oxidase type B, and the drugs that do that would be selegiline and rasagiline. And again, because there is a paucity of dopamine in the brain, a mode of treatment would be to give precursors of dopamine, and we can actually give the precursor as a drug, which is levodopa. But because levodopa can also be activated into dopamine prematurely in the blood by the enzyme dopa decarboxylase, and that dopamine that is in the blood will produce the cardiovascular instabilities and nausea and vomiting by activation of the chemoreceptor trigger zone, we can inhibit the conversion of levodopa into dopamine by giving an inhibitor of dopa decarboxylase, which is carbidopa. Therefore, in patients who have moderate to severe Parkinson's disease, the mainstay of treatment would be the combination of levodopa carbidopa. Okay, so that would be the different classes of agents that we use to treat Parkinson's disease. Thank you for listening.